So in the third topic, we'll talk about the inflammation and soluble factors, okay? So basically, we'll talk about 5 different subtopics in this major topic, inflammation and soluble factors. We'll talk about the antimicrobial proteins. We'll talk about the role of eicosanoids in the inflammation, as well the effects on vasculature, the effect of these soluble factors and the inflammation as well. We'll talk about the complement as well. And we'll talk about the acute phase reaction, as well as secreted enzymes and proteins which have immune function. Generally, the inflammation, this is a rapid response system, is dependent on the circulatory system. The system is based actually on a number of factors, including the homeostatic equilibrium that rapidly respond to disturbance, and they start generating and use different complement protein and the clotting mechanism to tag and trap foreign substance. So initially, you need to understand that in order for the immune system to start working, the foreigner or the invader need to be tagged to be identified, and then need to be trapped to have the action of the immune system on that invader. The second is the vasodilation and the process of extravasation to flush sites of infection. In other words, if you have a cut in your skin, this will lead to recruitment, as we will describe this in details later. This will lead to recruitment of a lot of cells and mediators to the site of the cut, trying to fight all sorts of infections that might arise at that site. So the process of extravasation, that means cells that will migrate from the blood vessels away to the outside, to the site of the cut or to the site of the inflammation at the skin. This is accompanied by as well a vasodilation. So you have as well the involvement of the reticuloendothelial system, and actually these are the phagocytes which are located in the reticular connective tissue, and this can remove all of the debris that arise due to the processing of the invader by the immune system. The fourth component will be the chemical identification system of the pattern recognition receptor. To be more familiar with the pattern recognition receptor, you need to understand that any invader have like a signature on its surface. These signatures help very much the immune system to identify this invader by different kinds of receptors. Generally, the major receptor that can identify this signature or the patterns associated with the invader is one of the major receptors called toll-like receptors. This receptor, including the toll-like receptor, can identify PAMPs and DAMPs. So what's PAMPs and DAMPs? Actually, PAMPs is a pathogen-associated molecular pattern, molecular signature of this pathogen, while the DAMPs is a damage-associated molecular pattern. So once this pathogen got damage or do some damage, these damage-associated molecular patterns can be recognized by the receptor. Hence, it will understand that something wrong, something is trying to invade, something is damaged, and will try to activate the immune system. So you can see here some examples of PAMPs. You can see even bacterial DNA, mycobacteria, gram-positive, gram-negative, even yeast. All of them can give rise to a lot of different PAMs. So these like the molecular signatures of these kinds of the invaders. So you can see even the lipopolysaccharide, lipoprotein, peptidoglycan, which exists on the membrane of the bacteria, the CpG island, which is a sequence of CG, CG, CG in the DNA of the invader. These representative examples of PAMs that can activate the toll-like receptor or this kind of receptor to activate the immune system. Hergatin. So after this brief introduction about the processes that contribute to the inflammation, as we said initially, we'll talk about 5 different subtopics. We'll start with the antimicrobial proteins. So actually the antimicrobial proteins, it is a collection of peptides that range between 12 to 50 amino acids. The net positive charge, okay, that disrupt membrane integrity. And they have, what I mean here, they have net positive charge that help to disrupt the membrane integrity of the invader. Many of these antimicrobial proteins are stored in a special type of leukocytes called neutrophils. And you can see here the table, a list of some of the antibacterial, very famous antibacterial compounds. You can see the name of the peptide, the site of action, the properties of the peptide, and the examples as well. So you can see collectins occurs usually extracellular, general extracellular. Defensins occur extracellular in the amniotic fluid, and of course intracellular, that's mean the site of action. The third type of the antimicrobial peptide, which is a cathepsylacidins, which actually it's the lysosomes of the polymorphinuclear leukocytes and macrophage, while saposin, it works in in the CTL, cytotoxic T lymphocytes, and the natural killer cells, while the mucins, it exists in our saliva. And this, you can see the properties, and these as well specific examples of these general classes of the peptides. For the second player of the inflammation, it's a collection of molecules which is called eicosanoids, and its role in inflammation. So these molecules are a family of very powerful hormone-like compounds, and they are produced in the body from essential fatty acids. Majorly, the main function of these molecules is to increase the fluidity of the cell membrane, and as well they act as a gatekeeper on the plasma membrane. So they have a gatekeeping abilities, and of course the gatekeeping, that's mean it will prevent the invasion of a lot of microbes and pathogens. They are membrane-derived lipids, of course, because they are generally from fatty acid, and they include prostaglandins, leukotrienes, lipoxins, and thromboxanes. And these are the general categories of eicosanoids, and they act as a potent paracrine and autocrine signals. And if you need to understand what is the difference between paracrine and autocrine signals, for paracrine signal, so this molecule will trigger and affect any specific nearby cells, while the autocrine signal, these molecules will affect the same cell which secrete this molecule. So this is majorly the difference between paracrine and autocrine. So these molecules as well is responsible for many of the beneficial effects of the good fats. However, some of these molecules, such as those derived from arachidonic acid, are potentially harmful if excess amount of these arachidonic acid build up in our body. So there are three families or series of eicosanoids. Series 1 and 3, series 1 and 3 are generally good ones, while series 2 is potentially harmful. Phospholipase A2 can release arachidonic acid from the membrane of phospholipid, which are central to produce the eicosanoid. So we have a major player here, the phospholipase A2, that help to release a lot of eicosanoid from the phospholipids in the membranes. So generally, these molecules are very good inflammatory endogenous mediators, and they can activate antigen-presenting cells, APC, antigen-presenting cells, and of course, particularly dendritic cells, which induce, as we said before, which in turn will induce the adaptive or the acquired immune response. As well, these molecules play an important role in connecting or crosstalk between the innate and the acquired immunity by acting on cells of the both system of the innate and acquired. They regulate the vascular tone, they regulate the smooth muscle contraction, they play a significant role in the anaphylaxis and asthma, and they are primarily responsible for the 4 signs of inflammation. So actually, maybe you are familiar with these signs of inflammation, which include rubor, redness, include swelling, forming tumor, which actually rubor tumor, and dolor, the pain, which follows the redness and the swelling, and as well the color, the heat that is generated due to the lot of the metabolic action and the release of the heat at the site of the infection, or even if it will be systemic in the cases of fever. So these actually the 4 signs of inflammation, and the primary molecule responsible for these 4 signs of inflammation is these molecules, eicosanoids. And of course, these 4 signs will lead to the loss of function of the organ or the system that is affected by inflammation. Again, you can see that inflammation can arise from the action of these 4 inflammation sites: heat, redness, swelling, and pain, which ultimately lead to the loss of function. So when faced with invading microbes, the inflammation is our body system is trying to protect itself through the innate the innate system initially. So they develop 2 kinds of attack: the humoral immunity and cell-mediated immunity. So we'll talk in details in the coming lecture. So generally, the humoral immunity, it is the immunity which deal with molecules, while cell-mediated immunity, it's immunity which deal with cells. So that's the major difference between humoral immunity and cell-mediated immunity. And generally, we have for the innate and for the acquired, for both of these 2 types of immunity, each of them we have humoral and cell-mediated for both these major classes of the immunity. So how these molecules, the eicosanoids, is produced? So you can see here, it starts from the plasma membrane, as we have just said, under the action of the phospholipase A2, the arachidonic acid will be digested into a major molecule, which is the prostaglandin G2, that it is highly expressed in cases like pain and edema. And this actually, that's of course leads to pain. And of course, one of the medication is used in this case is aspirin, ibuprofen, indomethacin, and this can inhibit the release of this molecule. And that's why we will not feel pain when taking aspirin and ibuprofen in some cases, because this molecule is not produced. In other cases, it as well, this kind of glucocorticoid interleukin-4, which is a mediator from inflammation, will negatively regulate the production of this molecule, as well as interleukin-1 and tumor TNF-alpha, or tumor necrosis factor alpha, which is a growth factor, will potentiate the formation of this molecule. And all of these molecules, actually we'll talk in details later, and these are mediators of inflammation. Another action of the phospholipase A2 on the arachidonic acid through the process of lipooxygenase is to form another important molecule, is the leukotriene A4 hydrolase. And this molecule, it leads to smooth muscle contraction and cell infiltration. Of course, you need to understand that one of the major processes that occur in inflammation is a process called vasodilation and vasoconstriction as well, and the extravasation. The process of controlling the contraction of the smooth muscles of the blood vessels that is near the inflammation site will help to the process of extravasation, or the migration of the cells, of the immune cells, from the blood vessels to the site of inflammation, and so to do its action as an immune cell. Today we'll talk about effect on vasculature. So what inflammation do to the vasculature? The vascular dilation occurs immediately when stimulated by histamine and bradykinin and prostaglandins, and these lead to this dilatation leads to increased permeability. While the vasoconstriction, generally it is defined as the restricted flow, permits clotting as well, and confines pathogen locally, so to prevent it from spreading and have systemic dissemination. Mast cells is very important because they regulate the vascular tone by producing vasoactive substances, while leukotrienes stimulate a smooth muscle activity. Cytokines CCL8 recruit neutrophils, another important player in inflammation, to entry into the stromal site by the process we'll talk in details in the next few slides, is the extravasation. So you can see here, generally it starts by cutting of the skin. And you can see in the skin, there is the surface blood vessels having the blood and the phagocytic cells. As well, there are mast cells start to secrete the chemical signals, and macrophage as well. Soon after that, these cells start to migrate to the site of the wound, trying to engulf the bacteria or any pathogen that enters due to the cut, and they secrete a lot of chemotactic compounds that recruit more white blood cells into the site of action. Finally, as you can see that the macrophage, or the immune cells, by the process of phagocytosis, it engulfs most of the bacteria, and then the wound is now healed. So the inflammatory response attracts phagocytes to the injured area or infected area. A typical inflammatory response begins when an injury, as we have just said, that damages cells and allow bacteria to enter the wounded area. The wound damage plus the tissue damage plus the invading microbes provoke this inflammatory response. And this inflammation may result into an accumulation of a pus. And what's a pus? It's a thick, whitish mixture of dead bacteria and debris and living and dead leukocytes. So you can see here from the figure, it starts by tissue damage. that carry bacteria into the wound, the yellow bars. Then the wounded cells release, chemical compounds, the red dots, that stimulate the mast cells. You can see these green cells. The mast cells stimulate mast cells. Once the mast cell is stimulated, a histamine is released, which is the blue dots, and this histamine will increase the blood vessel flow as well as the permeability to enable the more phagocytes to be extravasated and go outside of the blood vessel to be recruited to the site of the wound and undergo phagocytosis. So this figure as well summarize the major inflammatory response at the wound. Other chemical released by injured cells initiate blood clotting as well, because you need to close the cut in the skin. And this leads to the clot plug up damaged blood vessels, reducing blood loss and preventing microbes from further entering into the blood stream. So let's see here more detailed figure showing how the cells are recruited or majorly the process of extravasation. So in this figure, we'll talk about the process of extravasation of the leukocytes from the blood vessel. So if you can see here, in the top of the figure, this is the lumen of the blood vessel. And you can see these are the endothelial cells. You can see around the blood vessel, you have two major types of cell. The mast cell, which actually made its main function owing to the chemokines or the chemotactic molecules they secrete, majorly they recruit neutrophils from the blood vessel. That's the process of extravasation. As well as macrophage nearby of the blood vessels, which mainly function in phagocytosis and cytolysis, will help in recruitment and as well in destroying and phagocytosis of the bacteria through the secretion of cytokines and a lot of ROS, which is reactive oxygen species. So how the process actually occurs and how the cells are extravasated from the blood vessels. So you can see here, initially the mast cell and the generally first the mast cell, due to the recognition of PAM, which is a pathogen-associated molecular pattern, this promote the mast cell to secrete these three types of compounds or molecules: histamine, leukotrienes, and chemokines. So what happened to the neutrophils? Generally, the neutrophils is streaming in the blood vessel, and sometimes it got attached to one type of a glycoprotein that is on the surface of the endothelial cells, which is called E-selectin. So you can see that E-selectin traps the neutrophils, which is moving in the blood vessels. The trapping of these neutrophils these neutrophils is trapped by another molecule, which is P-selectin, which is recruited to the surface of the endothelial cells through process of exocytosis. So once the neutrophils trapped or got hooked by these P-selectin, this leads to the activation of another glycoprotein, which is the integrin. See, this form represent an inactive form, and this form represent an active form. So you can see, due to this stepwise movement of these neutrophils over this red, yellow, blue glycoprotein, EP integrin, the neutrophils actually is rolling over the surface of endothelium. The integrin activation, which is the blue one here, and once the neutrophil attaches to this integrin, activated integrin, it will stop rolling and stop moving, and it will start to squeeze itself between the two endothelium to get extravasated and to be outside of the blood vessels. Once the neutrophil crosses the endothelial wall, it goes to the space outside the blood vessel and near the wound, and it enters the site of inflammation. This is a very good figure explaining the process.
At sites of injury, infection, or inflammation, cytokines are released and stimulate endothelial cells that line adjacent blood vessels. The endothelial cells then express surface proteins called selectives. Selectives bind to carbohydrates displayed on the membrane of the leukocytes, causing them to stick to the walls of the blood vessels. This binding interaction is of sufficiently low affinity that the leukocytes can literally roll along the vessel walls in search for points to exit the vessel. There, they adhere tightly and squeeze between endothelial cells without disrupting the vessel walls, then crawl out of the blood vessel into the adjacent connective tissue. Here, leukocyte rolling is observed directly in an anesthetized mouse.
الفيجر ده حقيقي يا شباب.
The up and down
شايفين
movement of the
النيوتروفيلز معلم
frame
عليها.
is due to the mouse's breathing. Two blood vessels are shown. The upper one is an artery with blood flowing from right to left. The lower one is a vein with blood flowing from left to right. Leukocytes only adhere to the surface of veins. They do not crawl out of arteries. Some leukocytes are firmly attached and are in the process of crawling through the vessel walls, whereas others have already left the vessel and are seen in the surrounding connective tissue. When the blood flow is stopped temporarily by gently clamping the vessels, we can appreciate how densely both vessels are filled with red blood cells. Red blood cells do not interact with the vessel walls and move so fast under normal flow that we cannot see them. When the blood flow is restored, some of the leukocytes continue rolling, whereas all non-interacting cells are immediately washed away by the shear.
So what about types of inflammation? So majorly, and probably you knew already this, there are is classified into 4 types: either acute or the Lewis trip response, or chronic or type 4 hypersensitivity. For the first one, actually, it is exudative inflammation. That's mean a lot of excessive fluid. It is supportive of or purulent neutrophils, so these neutrophils go to that places. It is, you can see at the site of the inflammation there are serous clear fluid, and as well hemorrhage vascular damage. While for the second type, you can see capillary dilatation, arterial dilatation, as well as edema. While for the third type, you have a lot of noxious stimuli that is persistent, keep on attacking the body. While for the last type of hypersensitivity, actually, it is characterized by attempt to physically isolate the pathogen within the macrophage, and this characterized by large nucleated foam cells form as a syncytium of macrophage. So you can see like a big chunk of cells, which actually like a syncytium of a macrophage. So inflammation either can be local or systemic. The fever actually is a systemic inflammation response triggered by virogens, and a lot of virogens released by macrophage and toxins from the pathogen. While the septic shock is a life-threatening condition caused by overwhelming inflammatory response. So actually the inflammation, as you already know by now, it's a type of defense of against pathogen in our body. But it's overwhelming inflammation with these lot of cytokines and this lot of exhausting the immune system can lead to a life-threatening condition, which is a septic shock. So for fever, actually fever differs from everyday inflammation that might we have, that it combats large-scale infection. So if invader survive these defenses, causes a serious infection that may trigger fever, which actually important, very important, this systemic inflammation, very important in slowing down the microbial reproduction and enhance the body's own fighting abilities. And actually fever is controlled by the hypothalamus. And obviously you know already that the hypothalamus is the body thermostat. And certain type of bacteria as well as phagocytic cells that respond to the infection produce a very triggering molecules called biogens. And actually why it's called biogens, that's a Latin word that means fire producers, because these, which actually triggers the one of the PAMs that triggers the inflammation. These pyrogens travel in the bloodstream to the hypothalamus and raise the thermostat set point, and now the body responds with increased fat metabolism to generate more heat and constriction of the blood vessels supplying the skin, so to reduce the heat loss through the skin. And all of this as well accompanied by shivering, and these are the major signs of the fever. So how does fever actually combat infection? An elevated body temperature will provoke the increased activity of phagocytic cells, and as well will decrease the reproduction and the division of some types of bacteria. As well, it will increase the multiplication of cells of the adaptive immune response, the T cells and its the T cells and its sisters and brothers and daughter cells. And as well, it will stimulate this fever, and this elevated body temperature will stimulate virus-infected cells to produce a very important molecule, which is the interferon, that increase the resistance of other body cells to the viral attack. The interferon as well stimulate the NK cells to destroy virus-infected body cells. Extremely high fever may cause brain damage. We'll talk now about the complement. So the complement, actually, it's a family of interacting proteins, more than 30 types, and produced by the liver and released into the blood. And that's normally what happens inside our body. Once it's triggered, these complement, they follow cascade of reaction to attack foreign substance, and they can deposit on the pathogen surface and triggering the process of phagocytosis and inflammation, and as well pathogen lysis. The recognition activate a sequence of caspases that generate the anaphylotoxin, opsonine, and membrane attack complex, MAC. So all of these will lead by the end to the destruction of the pathogen, or destroy the pathogen. So in this figure, we can see what are the different types of complement. So actually complement move through 3 different pathways. You can see the lectin pathway starts, and you can see it starts with C4, which is complement 4, that activates C2, activates C3, and then after that C3 is cleaved into 2 other components, and then the C5 and so on, until formation of the membrane attack complex that is formed on the surface of the pathogen. So you can see here that you have a classical pathway that starts from C4 by the activation that involves the activation, its activation involved in must be involved by antibodies, while the alternate pathway starts from C3, and its activation involves C3 and C3b and the reactive oxygen species. The classical pathway is considered to be adaptive immune response since there is involvement of the antibodies. While the other two ways, the lectin and the alternate pathway, is considered to be related to the innate immune response. All of these complement protein will lead by the end to the process called opsonization, one of the major process, which is like a coating of the antigen. And in some cases, you will see that it will form a membrane attack complex. And this actually, the component of the membrane attack complex, actually, it's the C6, 7, 8, 9, will form like a complex over the surface of the pathogen, and leading to formation of a pore that will release the internal content of the pathogen, leading to the destruction of that pathogen. As well, you need to know that C3 as well, it can be cleaved into C3a and C3b, as we have just said, C3 can be cleaved to C3a and C3b. But this figure shows as well that C3b can be cleaved as well into C3f and IC3b with the help of factor I. That this one can be further cleaved by factor I to form C3dg. And all of these can bind, and C3d can bind to special type of receptor on the surface of the pathogen, is a complement receptor 2.
Complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different foreign molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are
actually the process of opsonization. It is the process of coating of the pathogen by the complement or antibodies, or both complement and antibody, as we will see now.
Are C1 through C9, named in the order that they were discovered, not in the order in which they function. The complement cascade can be activated by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated
as we have just said, as we have just said in the figure, this pathway is activated by the involvement of the antibody.
C1 then cleaves C2 into C2a and C2b, and C4 into C4a and C4b. C2b and C4b combine to form a protease called C3 convertase.
And of course, C3 convertase is a very important enzyme, likewise C5 convertase, that each of them will cleave the corresponding complement protein. So C3 convertase will cleave C3, and as well C5 convertase will cleave C5 into C5a. And you can see important thing here as well, that actually that C3 convertase, actually it is a combination between C4b and C2b, as you can see from the figure, the animation.
And C4b combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3a and C3b. In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3a and C3b are constantly being formed from C3, but without activation, they are soon destroyed.
So that means that C3b, once formed, it waits for an antigen for activation. So if it's formed and there is no antigen, the C3b will be destroyed because there is no need for that complement.
C3b reacts with the proteins factor B, factor D, and properdin to form a complex called C3 convertase, which cleaves C3 into C3a and C3b.
This you have seen that this differs from the other C3. It induces more the formation of more C3A and C3B.
Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3A is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3A and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have
Here you can see the process of opsonization. The binding of C3b on C3b on the surface of the pathogen will facilitate the process of phagocytosis, while C3a will induce the process of inflammation.
Binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properidin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5A and C5B. C5A enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components, including C6, C7, C8, and C9 to form a membrane attack complex.
While C5b
this structure forms a hole in the cell membrane and causes cells to lyse.
Attack complex. So you can see here that the complex formed a pore on the surface of the membrane of the pathogen, leading to the release of the internal content of the pathogen, and eventually will lead to its destruction. So this figure will summarize the different function of the complement system. So in the first function, you can see that three components involved in the recruitment of the inflammatory cells and anaphylatoxins, which are C3A, C4A, and C5A. While the second function is the opsonization of the pathogen, and it only involves here the C3b can all coat the bacteria, and it can as well the clearance of the immune complex can be formed by the binding of this C3b with the antibody to form immune complex, which is important as well, as we will see later in the process of coating as well. So we have recruitment of inflammatory cells, and we have opsonization of a pathogen, and the third function actually we have just said, which is the killing of the pathogen through the formation of a membrane attack complex that forms holes in the membrane of the pathogen. And the major complement which activate this one is the C5b. Again, this is the three different pathways: classical, lectin pathway, and alternative pathway. And the involvement of the first one involved the antigen-antibody complex, while the second one involved lectin binding to the pathogen surface, while the third one is activated by the pathogen surface only. And upon the effect of C3 and C5 convertase, this lead to the complement activation, which give rise to all these molecules, and each of them is involved in a different mechanism in killing or removal or killing or destroying of the pathogen, either by the recruitment of the inflammatory cells and subsequently the release of a lot of chemokines and cytokines and leukotrienes and histamines, or either by the opsonization, or either by direct killing by formation of the membrane attack complex. So the alternative pathway, actually, it's more primitive. It is the most primitive of the pathway because it is initiated by just simple attraction of the early factors to the surface of the microbes. So bacteria poly and lipopolysaccharide on the cell envelope of gram-negative bacteria, they serve as a potent stimulator to this complement system. The complement receptor on the dendritic cells in lymph node collect debris for displaying and screening by mature B and T cells. Any deficiencies lead to bacterial infection. The loss of the C3 function leads to general bacterial infection. And you already understood now that C3 is a major complement in the 3 different pathways. While the loss of C5, 6, 7, 8, 9 leads to a susceptibility to meningitis. You can see here from the table, it summarizes the receptor needed for the different complement ligand. So you can see that for C3b, 4b, and IC3b, it needs a specific receptor, CR1, while the different other receptors, CR2, 3, 4, 5a, and 3a, all correspond to its specific ligand. And you can see the examples of the cell type that have these types of receptor, either B cells or phagocytes. So in this table, you can see that how important antibacterial action by these serum complement. So the main function, actually, as you understood from the last few slides, the main function of these serum complement is recognition. So these complement proteins can recognize the pathogen, can recognize the invader, and this recognition through the 3 different pathways, either classical or the lectin or the spontaneous pathway, which actually the alternative. So you can see that it describes how this pathway recognize either non-specific, either by sugar binding protein, either by the natural antibodies, either by binding to the antibodies for adaptive and triggering of the adaptive immune response in case of classical. In case of alternative, you can see this recognition requires some host proteins to prevent the self-recognition. The second function of the complement, serum complement, is the convertase, because as you have just seen as well from the video, that it can form some reactive proteases, which are the C3 convertase and C5 convertase. These 2 molecules is very crucial in the continuity of the 3 different pathways of the complement. While the third function is the anaphylotoxin, which is actually a potent stimulator of phagocytosis. If you remember, the first function of the complement is the recruitment of the inflammatory cells. So how recruitment occur? Because it recruits under the function of 3 major complement, which are C3a, C4a, C5a. Going back to this table, to this summarize, you can see here that these are the 3 same molecules, C3a, C4a, C5a. So these, the release or the formation of these complements will help in the recruitment of the inflammatory cells. As we have just said in the first part of the lecture, once these cells are recruited, they start to secrete a lot of factors that leads to inflammation. The fourth function as well, you already have been told that the opsonization function, as we said, that it can coat the surface. And this opsonization, either as you have just said, C3b coating the whole surface of the bacteria for enabling or facilitating the process of phagocytosis, or either this C5b directed the formation of MAC. What's MAC? It's the membrane attack complex. So actually, this opsonization C3b or C5b, which is described here in that figure, You can see C3b coating the whole bacteria, while C5b activate C6, 7, 8, 9 to form the membrane attack complex. So as I just said, so this table explain what are the important function, or actually the antibacterial function by serum complement. The last function as well is permeabilization, because actually when the membrane attack complex is formed on the membrane surface of the pathogen, it leads to increased permeabilization that lead to the leakage of the internal content of the bacteria to the outside, and it is important in destruction of that invader. So let's simplify the process by looking to this image. Assuming you have a dinner and this is the turkey, so actually this is an antigen. This is an antigen. If you eat this antigen straight, so you eat it straight, so that's non-opsonic phagocytosis. So you ate this antigen without the help of a coating material on the antigen. But in case if there is antibody, which for example, let's say fork, the fork you used to eat. If you use the fork and you hold the turkey, which is the antigen, and you ate it, so that's actually the opsonic phagocytosis. Another type of opsonic phagocytosis, when complement is attached to the antigen, when complement is attached to the antibody, this is another type of opsonization that facilitate more the phagocytosis. So let's talk about the acute phase reaction that occurred during inflammation. So actually it resulted due to acute phase proteins, which are these acute phase proteins, its concentration increases in the plasma. And actually there are two types, the positive acute phase protein and the negative acute phase protein. And both of them coordinate in response to inflammation. The decrease the synthesis of such protein is to save the amino acid for producing positive acute phase protein more efficiently. So the decrease in the synthesis of the negative forms. These decrease in the negative APPs can be used as a marker of inflammation. So in a test, if you wanted to, in a clinical lab, if you want to check for inflammation, you can measure the concentration of the acute phase proteins, especially the negative acute phase proteins, and you can indicate, or you can say if there is if there is inflammation or not. One of the major famous positive acute phase protein is the CRP, which is a C-reactive protein, and the complement, of course, complement factor 3, which is C3, and as well fibrinogen and hepatoglobin. The major negative acute phase proteins are the transferrin and albumin. The liver compensate for the shift in protein content in these acute phase proteins. So actually in a process of fast acting and non-specific response. So you can see here, if there is a local acute inflammatory response, that leads to the secretion at the site, interleukin-1 and tumor TNF-alpha and interleukin-6. Once it's sensed by the liver, these lead to the formation of acute phase proteins, as we just said, like C-reactive protein, as well as mannose binding protein. So once interleukin-1 and TNF-alpha and interleukin-6 are sensed by the liver, the liver starts to produce acute phase protein, such as CRP, C-reactive protein, and the mannose binding protein. And if you remember mannose binding protein, it's the MBP, which actually it's activator of the classical pathway of the complement proteins. As well, the local acute inflammatory response, and due to these cytokines, will activate the hypothalamus to produce, as we have said earlier in the lecture, the hypothalamus to produce prostaglandins that help in the increase in the body temperature and will lead to systemic inflammation, which is a fever. As well as interleukin-1 and TNF-alpha will affect the process of leukocytosis, will lead to more leukocytosis in the bone marrow. That's mean because your body is needing more white blood cells. So the process is enhanced in the bone marrow. So you can see here, once the bacteria is inside the body, you can see that due to the formation of antibodies specific to that bacteria and due to tissue damage, this will lead to complement activation. This complement activation, if you remember, you have one of the major compound is C5a, and this 5a will induce the mast cell to secrete actually the chemotactic compound, which majorly is a histamine from the mast cell. So you can see histamine as well as prostaglandin as well as leukotrienes will affect the permeability of the blood vessel nearby to have the cells of monocytes, neutrophils, and some of the lymphocytes will exvasate outside of the blood vessel. And you can see that here as well how the cell is attached, each one of them by different selectin. As well, these bacteria will trigger the macrophage to do what it do actually, which is the process of phagocytosis. The addition as well of these interleukin-1, interleukin-6, and TNF-alpha, tumor necrosis factor alpha, as we have just said in the previous slide, will enhance more the process of extravasation. The release or the escaping of these neutrophils outside the blood vessels, and it will be recruited to the site of inflammation by a process of chemotaxis through the activation as well of C5a. So actually the last subtopic of this inflammation and soluble factor, actually we'll talk about the secreted. It's like a summarize of the different type of secreted enzymes and the protein which have an immune function. So first, of course, the famous one is the lysozyme, which is found in saliva, and this hydrolyzes or destroy or break down the polysaccharide on the bacterial cell surface. The phospholipase, which actually, if you remember, it shares its part in the formation of the eicosanoids because it breaks down the arachidonic acid. So it hydrolyzes actually the phospholipids into fatty acid and lipophilic substance and initiate as well the release of eicosanoids. While the lactoferrin, it's actually an iron-binding globular protein and is secreted by the exocrine glands and neutrophils. It's like once it is secreted, it's called serum lactoferrin. And they actually measure, its main function is to deprive the bacteria of iron. And of course, iron is important in the process of division of the bacteria and the metabolic process that occur inside the bacteria. While the lactoperoxidase, actually it's a bacterial oxidoreductase, and it is secreted from the mucosal glands and as well salivary glands and mammary glands. And it catalyzes the oxidation and the production of water using the hydrogen peroxide as electron donor. So actually, it removes the reactive oxygen species that present. While the myeloperoxidase, actually as well, it's a bactericidal oxidoreductase, and it is expressed in neutrophils and stored in a sort of granules which is called azurophilic granules, known as the primary granules, and secreted following activation. So these myeloperoxidase peroxidase is stored in these types of granules, and once the neutrophils is activated, it releases this enzyme, which is a bactericidal oxidoreductase, which actually do the same function of, because it's oxidoreductase, as lactoperoxidase, but it's a different source. The other one, lactoperoxidase, originated from the mucosal glands, but this one originates from the neutrophils. And actually, as well, it catalyzes the chloride ion oxidation using hydrogen peroxide to form hypochlorous acid. The catalytic factor is the heme, which is the iron, that gives the green color of the pus. If you have a wound and you have a pus and it's green, you understand that there is a myeloperoxidase that worked at that site of inflammation. While xanthine oxidoreductase, which is the last one, is as well oxidoreductase, but it converts hypoxanthine into xanthine and hydrogen peroxide and water, which is further broken down into uric acid and water. So it converts the aliphatic compounds to aliphatic alcohol and two superoxide radicals of oxygen. And of course, this will burden our body by the formation of a lot of reactive oxygen species. That's why we have a lot of antioxidant mechanism in our body, and that's why we need to eat a lot of green vegetables, and a lot of especially like red grapes, which have like a lot of antioxidants, to try to remove these lots of reactive oxygen species that is already generated due to the activity of our immune cells and the immune response. And you can see here the xanthine oxidoreductase. Actually, it's a enzyme that participate in our body in the immune response or the defense of our body. And it can generate reactive oxygen species like the two superoxide radicals of oxygen. And if this reactive oxygen species is left in our body without being removed from our circulation, actually it will destroy all the biological molecules that we have, proteins, lipid, and carbohydrates, even the DNA, even the RNA. So that's why we need a lot of antioxidant to remove these normal byproduct, which is actually harmful to our body. So this type of enzyme actually is widely expressed in tissue and is major constituent of fat globules in breast milk. And this concludes our third topics, which is inflammation and soluble factor.